Today on Immuno-Tea Time, we're joined by Dr. Miriam Wöhner, a junior group leader and immunologist at FAU Erlangen. Her work explores how immune cells like B cells and macrophages shape tumor growth and metastasis, using tools like CRISPR-Cas9 screening and mouse models. She's also uncovered key transcription factors that guide plasma cell development and recently identified a new B cell subset with potential anti-tumor activity. Miriam, we're so excited to have you. Let's sip into science.
Thank you so much for having me. It's really nice. It's a really nice setup, and I really like your podcast, actually.
Thank you very much. We are drinking today normal black tea, and so let's hope we also get a bit wake up, because today we are recording in the morning. So.
Yep.
Oh, you mean I'm alternating between coffee and black tea now? Yeah. Awesome!
Yes. So Miriam, could you talk about a bit about your path during? How did you start in biology, immunology, tumor, everything?
Yeah, sure. Well, actually, I didn't know that I wanted to go into the immunological path at all. Like, I came back from traveling New Zealand, Australia, and Fiji for a year, and I was like, what do I do with my life? I want to go to Thailand, but maybe I should try to do something a bit more serious with my life. And then I applied for biology. And for a moment, I was really, really, really sad that I was taken and couldn't keep on traveling. And then somewhere around the third semester, I realized that I really like biology and that I really enjoy what I'm doing. And then I had to choose in the fifth semester between molecular plant physiology and genetics. Mm-hmm. And for me, the molecular plant physiology was really compelling because that was the only topic in the whole studies where actually a woman would give a lecture. Like everything else, every single lecture was by men. So that was a very compelling argument. But then I decided that genetics is more of an interest for me. So I went to a man, to Professor Lars Nitschke, and I had an amazing time in his lab, and I decided to stay for my PhD as well.
Oh, so there was no female lecturers?
No, not that I'm aware of. Like everything, like every professor was a man. Even the female lecturers, teachers that we had were postdocs. Mm-hmm. It was really bad when it came to representation or to giving you a feeling that women in science can get anywhere, basically. And it was a bit sad.
What did you love so much about Lars's lab?
Oh, just the whole atmosphere. Like, I really loved that I could develop my own ideas, my own projects, but we were such a great bunch of people. Like always, I like Lars a lot, and he always was a great mentor anyway. Mm-hmm. He's a great mentor. But additionally, the whole lab with all the PhDs and everybody were just a bunch of friends. So it felt like, yes, I'm doing research, but I'm also meeting my friends, and that's such a great combination. And then I got some projects failed, some were really nice. I ended up with having 3 publications from the PhD. So science-wise, it was also really great time. Mm-hmm. It was super nice, yeah.
That's really good.
Yeah, you also mentioned that there were no women. Did you ever feel underestimated during your research, or?
Not necessarily underestimated. For me, it never really occurred in the beginning that I could end up anywhere as a group leader. Mm-hmm. Because it was always clear to me that this is some mutual exclusive thing to have children, family, and a scientific career. Mm-hmm. So it only changed when I afterwards went to Vienna. So I went to the IMP, that's the Institute of Molecular Pathology, which is an amazing place to do research. Plus, there were a lot of women, and a lot of women who had children. So there I saw for the first time, oh, you can have family and science. Wow. Mm-hmm. But here at the university, when I was studying, I didn't have the feeling that that was true at all.
Mm-hmm. Mm-hmm. Yeah. And how was I think in Vienna you were in Buslinger's lab?
Yes.
Yeah.
I was with Professor Meinrad Buslinger, who is one of the big immunologists. Mm-hmm. So he had this big lab, and we were doing basic research on B cells, and it was a completely different world. So I came there, and the IMP has a lot of funding. Mm-hmm. So I came there, I was like, oh, when I do my PCR, how much TAC am I allowed to use? Because here it was always a bit like, oh, and don't use more than 0.1 microliters per PCR. And I was like, how much TAC am I allowed to use? And this PhD student, he looked at me and was like, however much you want. I was like, whoa! So there the science is just very different. Mm-hmm. I mean, the whole educational purpose is not as much as at university, meaning we didn't have bachelor students. We had summer students, and we had now and then master students, but you would give no lectures. So you were super focused on your research, which is super great if you want to do really big research. Yep. But I actually like the aspect a lot here to also do teaching. I just like teaching. Mm-hmm. So the IMP was great to do big things, but I enjoy here that I can do science and teaching at the same time.
Cool. Yeah, that's nice. Yeah, also because you said that in the IMP there were like a lot of women with children.
Yes.
And we also had this thing that if you want to do research and you're a woman, it's very difficult to become like a PI because you have to take care of your children, to stay at home or whatever. So at the end, you are not doing that. So
yeah. So at the IMP, they generated an infrastructure which made it easier. Mm-hmm. Not easy per se, but easier. For example, there is a pregnancy lab. Mm-hmm. So when I was pregnant, I could immediately tell them that I'm pregnant because I was still allowed to work. I had to hand in my protocols, and they had to approve it, and I had to work in the pregnancy lab.
Wow.
But for most of the things, I could still do them, which is a big plus because you don't feel like you put yourself or the child at risk, and at the same time, you can still do science. So that was a big plus. But then, for example, when I came back, I asked, I'm still breastfeeding my child, where do I pump? And they were like, well, you can use the toilet. I was like, the fuck? I mean, what kind of answer is
the
that?
Toilet.
It's like the best place to relax, take care of that. Also, when I was pregnant, there is this Austrian law that you're allowed, or that you are supposed to have a place to lie down during the day if you're not feeling well. So I asked for that place, and they said, you can pick between the couch in the study room where there were like 200 desks, or you can go to the 5th floor and use the couch there. But on the 5th floor was the head of the IMP, then the second director of the IMP, Meinrad Huslinger, and like the whole HR. I was like, I'm not going to lie down there on a couch. So there is still room for improvement, but it made it way, way, way easier that there was the pregnancy lab. Plus, one of the new junior group leaders there was pregnant at the same time as me. And she built a lot of infrastructure. She was like, we need a seminar room with toys. Mm-hmm. I want to give or hold lab meetings with my people, and I want to bring my child.
Wow.
So that was great, and that helped a lot.
Mm-hmm. Yeah, I mean, we have known for years that it's been better and better for women, so it's good to hear that it's happening.
Yep.
Yeah, at least at those places. The other thing is that the IMP has its own kindergarten. Mm-hmm. So here my children are at kindergarten, but the first one I had to pick up the kids at the latest at 3 on Fridays or 2. So are you supposed to work full time with those hours? Then there is a lot of closing times. Just the regular ones when they just have 3 weeks in the summer, 2 weeks here, 1 week here. Then they have a lot of days in between. And then, of course, the kindergarten just randomly shuts down because there's not enough people working there. Mm-hmm. At the IMP, they pay their own kindergarten. It has extended opening hours. Mm-hmm. It doesn't have any times where they are just not there. And it's substituted by the IMP. So you only pay €100 in contrast to 400 or 500 euro here per month.
Yep.
Mm-hmm.
So that alone is such a big plus.
But that was in the IMP. Do you think in academia things have gone better since then?
I mean, there is kindergartens here now belong to the university. That helps a lot. There is also some programs like when you paid by the big TRRs from the DFG, so the Transregio, where several labs from different cities basically belong together. For example, there is a budget for if you want to attend a meeting that you can get a babysitter. There is money for just toys. So we have a box of toys here so that when I have to bring my kids, I can basically put them here in the seminar room and they can play and so on. So there is a different mindset happening. It's slow, but it is happening.
That's good.
So you said like things were, I don't know, reasonably okay at the IMP, but what's it like being a woman in the other parts of your career?
Yeah, so for me, I had some major roadblocks, I would say, just because I didn't fit into the time frame that was once set for your career. Meaning, after 3 years at the IMP, I had an extremely nice first author paper, and that would have been the perfect time point to apply for group leader positions. But I knew I wanted to have a family. So I was like, starting a lab and being immediately pregnant is not the best move. So I should have my children while I'm still a postdoc. Now, stuff doesn't work like that, of course. So it took me 2 years to get pregnant. And that, of course, leads to you falling out of all the categories for a lot of the funding. For example, if you want to have an early researcher funding at the DFG, your scientific age, meaning the time that passed from your defense, can be a maximum of 4 years. So immediately out of the picture. Now they started to adapt things that you can subtract 2 years for every child. Meaning if the scientific year can be a maximum of 7 years, and I had 2 children, I could, in theory, subtract 4 years. But that's not a reality because I did research in between, of course, but I lost 2 years for getting pregnant, then I was pregnant, then I was on maternity leave, so the 2 years don't cover that at all.
Yeah.
And then you fall out of a lot of funding. Then the second thing that happened to me was that I got pregnant with my second child and my contract was running out. Because in science, if you're not extremely lucky, your contracts just run out. I mean, I'm on the longest contract I've ever been on right now, which is 3.5 years. So before, I had 1 year, 2 years, another 1 year, and so on. So the contract was running out. Meinrad Buslinger was at that time point shrinking his lab because he was 69 and he had to slowly start about retiring. So I went back to Germany and got an unemployment money. I got 250 euro unemployment money, which is great. Can make a total great living out of that. Plus I was pregnant, so try to find a new job in a lab with being pregnant. I mean, why would anybody hire a pregnant woman who can't enter the lab, basically? So that was not great. And that basically cost me another half a year. Then afterwards I was like, okay, I can work again. Let's try to get a group leader position. And then you apply for group leader positions and you immediately sort it out because you don't have third-party funding. Okay, let's get third-party funding. Oh, you can't get third-party funding because you're not attached to a lab. So you end up in this weird place where you go like, I'm in the void where people are not supposed to be, but I ended up in this void. I'm probably not the only person in the world, but there is no real plan or blueprint of getting out of that. So what happened was that I went back to being a postdoc, basically. So I applied with Professor Falk Nimmerjan, who I knew from my PhD thesis, who had a really nice project working with the macrophages and the metastasis, basically. So trying to find a solution or to help cure metastasis in the lung by defining the macrophages, a specific cell type. And I went back to that, and now I'm doing the habilitation, which is a very, very German thing, which is kind of the equivalent to a junior professorship. But this was like so many steps that you had to take back in order to get back into the system. And as I said, everything is measured in your scientific age, and my scientific age is too old for the position that I'm in, but I'm not the only person who at that age range tries to become a group leader, and our system is really screwed there.
But it's really hard when my female classmates ask a professor, like, is it possible to have a family and have a career in research? The professor's like, yeah, it's totally possible. As soon as you become a group leader or a professor, then you can have kids. But it's like,
yes.
So how do we? I feel like we're missing the middle part. Like,
Totally. Plus, it's not always, you can't time your pregnancy.
Yeah.
Like for a man to say that, yes, it might work out. But for a woman to find the perfect time point, and you feel your, as stupid as it sounds, your biological clock ticking, you know, like, oh, damn, I'm now mid-30, how many years will it even be possible? I want 2 kids, I need to have some time in between, obviously, but how is that supposed to fit with the schedule of when am I supposed to be a group leader? So it's planning your scientific career and planning a family at the same time is still hard. Had I been lucky and just gotten pregnant immediately, I think things would have worked out completely different, but that's not
Maybe
how
I
nature
should get
is.
pregnant now.
so then what's the advice you would give to like a young woman in science? So, someone who wants to have a family. Knowing what you know.
I still think that having your child during the postdoc is the best time. But so in hindsight, I would have been smarter to really go for the group leader position and say, screw it. I mean, if it comes to the worst, yes, I'm letting my lab down for a few months. But it would have been the smarter move to put the career step before the family luxury step. But then again, it's also super stressful to be a group leader and then saying, hey, I'm running around like a maniac nonstop anyway, but now's the perfect time to have kids. I think it's really hard as well. There is no perfect time point. Maybe during your PhD if you feel ready, but that's a different story.
We should interview someone who had a kid during their PhD.
I don't know anyone, so yeah. Okay, so now let's come into topic. So you have worked in B-cell biology, in macrophages, transcription regulation, tumor immunology. There's a lot of different points. What brings it all together?
So I actually started off with working with tumors in Professor Nitschke's lab. So I had this mouse model where we tried to target B-cell leukemia using a sialic acid. That's a sugar. So on the B cells, you have a very specific surface protein, which is called CD22, and that binds sugars. So we thought if we make a sugar which can only be recognized by CD22, meaning only by the B cells, we can kill specifically B cells. Because when you have leukemia or lymphoma with B cells, often what you get is rituximab. Rituximab is an antibody which binds to CD20. But what happens is that the B cells, they don't require CD20, and often they just shed it, meaning they get rid of CD20. So we thought it would be good to have an alternative therapy. Let's target CD22. And we had this immunotoxin, meaning this sugar linked to poison, a toxin. And it worked really great in tissue culture. So when I applied it to cultured B cells, they would basically all die. Then we decided let's go a step further and try it in a mouse model. And what happened is that we could cure some mice, but it had ginormous side effects. So there was internal bleeding, for example. So they were like, no way, this is going nowhere, and we basically dropped the whole project. which is, the purpose of doing animal experiments in the first place, to detect whether something is toxic before you try it in humans, right? So the tissue culture itself was so promising, but no way this could have ever been used in human. So I decided, no, I never want to work on tumors again. It's too much of a hit and miss. So I was super happy when I came to Professor Buslinger's lab, because it was basic research, meaning a whole step further. So the idea was, again, B cells, and maybe I should give some context. So B cells are the cells which will, once they are activated, turn into plasma cells or memory B cells, and will provide the body with the antibodies to protect us from different diseases. And quite often, those B cells lose control, just because during the step where the B cells try to produce antibodies which are as perfectly binding as possible, they on purpose introduce mutations to the genome. This is a super risky step, of course. Usually the body does everything to avoid random mutations, but that's what a B cell does on purpose. And there is millions of B cells which at any given time points on purpose mutate themselves. So the chances that they do something wrong and lose control are super high. Luckily, we have a lot of regulation mechanisms and so on. So obviously, we're not all running around with having B-cell leukemia, luckily. But they are prone to that, not to the disease, but to doing stuff wrong just by their nature. Mm-hmm. So the other things that they do is they're supposed to produce antibodies, but those antibodies are not allowed to recognize anything self, meaning any structure which is in your body. And that's, of course, another thing which can easily go wrong. Mm-hmm. So targeting B cells can have there's two diseases where you can benefit from targeting B cells. One is autoimmunity, and the other one is B-cell leukemia. Mm-hmm. So that was the whole approach that we had in Professor Nitschke's lab, and now in the Buslinger lab, we were a whole step further in the process. What makes a B-cell a B-cell? What makes a B-cell lose control? How is a B-cell regulated? So really not with the purpose of targeting B cells, but just understanding what is going on in a B cell to actually develop. And I really like that, because basic research, it sounds, when you're just out of your studies and you want to cure everything, and there's this disease and you want to be the one to find the medication. Basic research sounds super boring. But it's so interesting when you're really into science, because you get to work on a specific project, and it doesn't matter what the outcome is. It's not the hit and miss, it's understanding. It's understanding what's going on in the cell. There is no right or wrong. And there is a lot of really important points there. And for B cells, they are tightly controlled by the so-called transcription factors. Those are proteins which bind to the DNA and decide whether a gene is even expressed or not, meaning they switch on genes, basically, or switch them off. And in B cells, there's very specific ones. There is PAX5, that's the transcription factor that Meinrad Buslinger discovered, which actually put him on the A-list of the immunologists. But there is also some other ones which are super important for the step between I'm a B cell or I'm a plasma cell which produces the vast amounts of antibodies. And I was super interested in what is regulating the step, what decides whether you are allowed to be a plasma cell. And that's what I was working on actually for then almost 8 years.
Very interesting.
Yeah.
So the difference between B cells and plasma cells is mainly that the plasma cells produce more antibodies and that there is this activation of this transcription factor?
Yes. So a B cell is there and is ready to do things. Mm-hmm. So now you get vaccination, you step into a rusty nail and somebody coughs in your eye, God knows what, you are exposed to antigens. Antigens are the things that can be recognized by antibodies. So a B cell will see an antigen, then goes to a whole process which is called a germinal center, where it meets basically cells which tell the B cell, hey, you're doing good. You're the one who has a really nice antibody, you're the one who's efficient, and at the same time they also select for, oh, you don't recognize self, because that's the ultimate fail if they recognize a body-owned structure. So when the B cell passes through all of that, it's allowed to become a plasma cell. And then a plasma cell is huge compared to a B cell, and it produces tons of antibodies. So that's where the actual adaptive immune system kicks in. That's also why you have a huge delay between when you're vaccinated and you have really efficient antibodies. I mean, your whole immune system, of course, regulates, or your immune system really early kicks in, but the antibodies, which really efficiently do something against the disease, they take days or even weeks to fully mature and really do something. That's why you get booster vaccinations and so on, and why you should be protected when you're vaccinated against measles, because the B cells are there. They remember that they've seen the antigen, and they are the ones who will constantly kind of provide antibodies and also detect if the antigen comes back.
So we have also talked now that we have these transcription factors that will activate the B cells that will change to plasma cells and so on. And we have also said that there are some antibodies that will recognize self-antigens.
Yes.
There are some transcription factors that can affect them in this process.
So the transcription factors per se are, of course, regulated as well. So when a B cell detects an antigen, there is a whole signaling cascade going into the cell, and then the transcription factors get activated. The transcription factors regulate themselves, plus there's several other layers of regulation, just how densely the DNA is packed, for example, whether the transcription factor can even attach to the DNA or not. But in principle, a lot of transcription factors do really broad things. Like one of the factors that I worked on is XPP1. So producing antibodies means that the cell is full of RNA. So in a plasma cell, you have up to 50-60% of all the RNA in the cell is just for producing antibodies.
Well, that's a lot.
Yeah, that's a lot, definitely. And so there is all this RNA, and that causes a lot of stress for the cell. So XPP1 comes in, and XPP1 basically makes sure that the stress is relieved. Now, if there is no XPP1 or something is deregulated, the body or that the cell can't cope with those amounts of RNA, and that will lead to a process called RID, which basically means that it just destroys most of the RNA that is in the cells, just to, I can't cope, let's get rid of all of that and try again. So this transcription factor is essential for actually getting the plasma cells into a stage where they can actually produce the antibodies. And there's a lot of those examples, and it's quite exciting to actually work with them, because there is so many methods that you usually don't employ for anything else, where you, for example, can see where exactly on the DNA does my transcription factor bind, what does it exactly regulate, what are the effects if it's gone for days. But there is also now methods that we started to employ when I left, where you can get rid of a transcription factor and see effects within minutes. So you can say, I shut down the transcription factor and I just measure after half an hour and see what's going on. And as I said, it's hard to grasp if you never did basic research, but actually this is really interesting research for me.
Yeah, I can imagine. Like back in the day when they were discovering transcription factors, doing that sort of thing would be like such a breakthrough.
Yes, no, it also was amazing that we actually managed to do that. That was lucky at the IMP, because the IMP is such a driven institute. So there is no let's maintain status quo. There's no, we have always done it that way. There is only, we need to develop new methods. We want to be in the forefront of science. So the neighboring lab of Professor Johannes Zuber, they developed a whole method where you would put a little tag onto your protein of choice, and then as soon as you add auxin, which is a plant hormone, that protein would just immediately disappear. And that was the breakthrough method. And it was so cool to be at a place where science is so incredibly up to date and where they put such an effort into developing really new and really cool technologies.
And talking about new technologies, we have also CRISPR-Cas, right? And
Yes.
you work with it.
Yes.
And I think, okay, because we know, but I think there are a lot of listeners that 100% have heard about CRISPR-Cas in the news, but they don't have any idea about what is it. Could you please explain us now that you are expert on that?
Yeah. So what I specifically did, and I will come to the method in a minute, was so we knew about we knew about transcription factors, which are important for B-cell development. So we knew, for example, as I mentioned before, PAX5 super important. Without BLIMP1, you don't get plasma cells. So plasma plus are the pre-stage of the plasma cells. You need E2A, E22, and so on. So we had this list, but we were sure, or we were convinced during our research that there is more factors which kind of got hidden behind the big ones. And of course, if you want to really understand what's going on in a cell, you want to have a full picture. So we decided to just target everything that potentially could be important for plasma cell development. So meaning we wanted to have a method where we could, in individual cells, destroy one gene. And that's what you can nicely do with CRISPR-Cas9. Because you say, I want to destroy the gene BHLJ15. That's another one that I worked on. I know, it's kind of breaking your tongue. Let's call it by its protein name. It's called MIST1. So MIST1, so you know the genomic sequence, so you can detect or you can just check whether there is a place where you can put a guide RNA. Meaning you design a specific sequence, and then when you introduce or when you have Cas9 in your cell, it will use the sequence, detect that specific place on the DNA, and then insert a mutation there. Meaning, I designed for the screen, we wanted to target 3,000 genes. We decided to use 6 guide RNAs per gene. So we knew we needed 18,000 guide RNAs. And we wanted to have enough representation of those 18,000 genes to detect afterwards what we actually targeted. So we designed this massive approach where in each cell, one specific gene, which might be important for plasmablast development, gets switched off. And then to check afterwards who could still make it to a plasma cell and who could not. And then we could figure out everybody who could still make it to a plasma cell obviously had a gene targeted that was not important. And whoever got stuck in the process had a gene which is important, which you overlooked before. But this was a massive, massive, massive approach. I think it's the biggest experiment I ever done. It needed almost 2 years preparation time just to, get all the right guides and to set up the system. And you don't want to have 2 guides in the same cell because then you would switch off 2 genes at the same time, which you don't want. And I ended up having 2 massive days where I had 100 flasks in the tissue culture just with the cells for harvest. For reference, that's a lot. That's like 2 full incubators just with your cells. Yeah. And we had 3 sorters, meaning that's a machine which you can use to tell cells apart and separate cells on certain criteria. So I used 3 sorters for the whole day just to get my 1 to 3 million cells together that I needed for sequencing. That was crazy. But we did find a lot of new and interesting genes. Well, that was pretty cool.
Yeah. So the CRISPR is just basically you can choose any part of the genome that you want and then cut it out.
It needs to have certain criteria, so there need to be specific sequences there, for example. But yeah, in theory, you can switch off most of the genes. It was not always possible to have 6 guides, so 6 places in each gene, because if you have a very short gene, you might only be able to do 3. But for the genes that we wanted to target, most of them, we managed to design 6 guides, yeah.
But I didn't even know how, back in the day, you would have to, I guess, introduce another piece of DNA into those cells and then cut out the DNA in those cells and then hope that it matches and fits together, and then you have a part of DNA which doesn't have that gene that is now inserted into the genome of that cell, which is kind of inefficient, right?
So one of the major issues was how to get the guides, so the small pieces, into the cells, because B cells, they don't like to have just DNA put into them, to say it very bluntly. So what you need to do is you need to have cells which produce viruses which carry your little genes or your little guides, and then infect the cells, the B cells, and then you need to you get a, in this case it's called transduction rate, meaning cells which actually got the guide of, let's say, 10%, and then you need to select for the ones which actually got the guides. And you always need to check that if you put in a few 10,000 guides, that the representation of what you wanted to put in and what you get at the end is still the same, that you didn't lose 5,000 along the way.
Yeah.
But I think, it's to do something on that scale, I guess 20 years ago would have been like incomprehensible.
Yeah, it was really cool. Again, collaboration with Professor Johannes Zuber, who he set up the whole system. So we had the expertise in the house. He did some really cool screens with that. There was some massive papers of him where he actually used exactly that method. So we were super lucky that we could kind of piggyback it. But yeah, for each cell type that you want to target, you basically need to set up the whole system. But yeah, it was massive. It's like the one biggest thing I did scientific-wise.
And then what was the most interesting result you had from that screening?
So we found a complex that was never implied in plasma cell development before. But I supervised a PhD student, and then I left, and she finished her PhD not too long ago. I'm not entirely sure whether they're in the process of writing up that paper right now, so I won't spell it out. But that was super promising. So we had some nice factors. We then, of course, you have to prove afterwards that this is really, really important. So you create a mouse strain which lacks this specific gene, and then you analyze whether this is really impaired in the plasma cell development or not.
Okay.
And that's what the PhD student then did.
Yeah. So you found a lot of new stuff.
Yes, there were some nice genes there. It's always the problem that you can't nicely knock out everything in a mouse strain. So sometimes this works really nice. Sometimes you spend long time trying to create a strain, which is, of course, tricky when you want to do a PhD in 3 to 4 years, and then it takes 2 years and you actually can do your experiments. But yeah, no, there's some really nice things in there.
Yeah, and with these results, if we try to make them a bit more translational,
yes,
what could we do? So we know now that there are some transcription factors that are making us go or not go to plasma cells.
Yeah.
So what will happen? Because we know that plasma cells are involved in several diseases like lupus or something like that.
Yeah.
So what can we do?
In theory, the idea is always that somebody else picks up on your basic research and uses it for therapy, for example. But that was not our primary purpose. As I said, we were a basic research lab. The idea really is to just understand what's going on, to really put together all the little pieces, to be a kind of detective and just try to figure out who is controlling whom and who can you, in theory, influence to get the different results. But the beauty really is of basic research that it doesn't have to be immediately used to cure a disease. It's really understanding how things work. And I enjoyed it quite a lot because to me it feels a bit more like being a true scientist in the sense of you discover things and they are always kind of useful. Even if you find something where you thought it might have a big effect and then it doesn't, you learn something. I mean, I also enjoy what I'm doing now, which is to actually, against the promise to myself, go back to work on cancer. But there you again are in this situation where you have an idea, you test the idea, and then it turns out that this potential cure is not doing anything. And then you go like, what do I do now? It's super hard to publish. Hey, I tried to use this antibody to cure cancer, but I failed. It's not the most exciting story in the world.
Yeah.
And the chances of actually finding a new cure are not super big. So it's always more tricky.
Yeah, because now you move into metastasis, right?
Yes.
Now you're studying metastasis, that it's when cancer cells from part of your body are seen in another part of your body.
Exactly. So to me, I have two primary organs that I look at, which are both very prone to metastasis, which is the lung and the liver. And they are completely different in terms of, in the lung, you breathe in a lot of things which are potentially bad for your body, like the whole day. There is a constant fight of your macrophages and some other cells against whatever crap you might have just breathed in. So I'm very interested in those macrophages. The macrophages are basically the big eaters. So they are part of your immune system. They recognize stuff as non-self, and then they will just try to get rid of it. And in the lung, you have a very specific subset of those macrophages, which are the alveolar macrophages, which sit in your alveoli, so that's where the air comes in, basically. So, and what we are interested in, and then in contrast, the liver, in the liver, you also have very specific macrophages. The most important one for our tumor question being the Kupffer cells, which are macrophages which reach into your blood and try to filter out whatever you have in your blood, what they can detect as non-self again. So both of them are super prone to tumors, and we try to understand what is going on when you have a liver tumor in the lung, what's going on the other way around, and how can the liver help us treat or prevent metastasis in the lung, for example. And for that, we actually need to really figure out what exactly the macrophages do, because, again, I said there is the alveolar macrophages, but there is a lot of subsets. They seem to be vastly different at doing different things. So we try to figure out which ones are the ones that support the tumor, because metastasis need a lot of support. Basically, without macrophages, metastasis have a hard time growing, because the macrophages, if they decide to help the tumor by whatever mechanism, they will, for example, attract blood vessels. For a tumor to reach a certain size, it definitely needs blood vessels. On the other hand, if you just get rid of the macrophages, it's also super bad, because so they try to do that in human, that was not a beneficial therapy. So the macrophages can make up to 50% of a tumor mass. So it was very compelling to say, hey, let's use the macrophages to do something against tumors. So what we did is we now used a very specific medication that we give intranasally. And that actually, in half of our model mice, led to no to little metastasis growth in the lung. The other half is still not okay. I mean, there's no influence there. But it's really cool to see that you put something really like a nose spray into the nose and it actually has a benefit.
Yeah.
That's what we are working on right now. But as I said, this is super early stage of doing anything that comes even close to a medication. But it's actually quite cool research as well.
It's amazing. That could benefit people with lung cancer so much.
Yeah, I mean, at the moment it's preventive measurement. But then again, like if you have a primary tumor and you disturb the tumor, being via surgery or via chemotherapy or via radiation, that can really lead to metastatic cells leaving the primary tumor. So actually there was a time window where one could actually really do something useful with that. But as I said, this is super preliminary, but it's quite exciting.
So could you apply the same principle to the liver? Or do the macrophages have a different regulatory role?
So the liver needs to be targeted completely different. That's the next step. So in the liver, we were busy characterizing which macrophage subsets are there, who are actually the big eaters, who is again helping. And now that we have a nice picture together with who does what, the next step would be to see whether we find a difference on the surface of those macrophages that we can again target, like with the alveolar macrophages. That would be super cool.
Yeah, I think so as well.
Because if I would ask, like, if I would think, like, okay, macrophages are big eaters, why are they not eating the cancer if they are like the bad cells, right? Like, yeah.
Some of them do. But again, it also can help the tumor to grow if the cells that the tumor doesn't need anymore are cleared away, because otherwise they're just accumulating cells that they don't really need on the inside, which might take up nutrients and so on. So there's really a lot of interplay happening, and it's a very, very complex system. And it's not so easy to figure out who is the good ones, who is the bad ones. But that's what I've been trying to do for the last 2 years, actually.
And how does a macrophage know what's good and what's bad?
That is an excellent question. I mean,
Congratulations.
So the macrophages actually, and now we go full circle, get a lot of the instructions from other cells, like, for example, B cells. Because when the B cells secrete antibodies, so when they recognize something as bad, or as an antigen, they will secrete the antibodies. And those antibodies can then bind to the tumor cell. And then the macrophages recognize, there is a lot of bound antibodies. They have specific receptors, the so-called Fc receptors, or in our case, Fc gamma receptors, meaning that they recognize a specific branch of antibodies. So they will know, this is a target for me. And that's definitely something that's happening, which in the tumor case, we can use to our benefit. Of course, as soon as it comes to autoimmunity, that is one of the things that causes all the problems, because you have an antibody against self, and now all the other cells of the immune system, which carry Fc gamma receptors, we go like, there's antibody bound there, let's destroy that.
Yeah. But you are a B cell person, right? You work every time on B cells, and now you're working on macrophages. Did you find anything in metastasis related to B cells?
Yes, I did. So when I was I did my macrophage things, and I just decided to, whenever I do macrophages, let's stain for B cells on the side. Just because I really like B cells. So I routinely did my B cell check in the metastasis and realized at one point that there was B cells, which I first found them in the lung, but they are all over the body basically, which don't fit a normal B cell description. So usually a B cell is supposed to secrete a lot of antibodies and thus activate other cells. But in recent years, there have been more publications coming up that B cells might also inhibit other immune cells. It's not entirely clear how. And I realized that there's a very specific subset, which is always there in our body, which expands as soon as there is a tumor present. So what those B cells are doing is they inhibit the other immune cells from doing something against the tumor. And actually, we are working on that right now because we can nicely characterize those cells, and we can show that they are full of different components which they can use to signal to other immune cells in the not so conventional B cell way, like specific interleukins and so on that were not supposed to be in B cells. So we actually think if we target those specific B cells, it might be very beneficial to prevent or to inhibit the tumor growth by getting rid of the cells which stop the other immune cells from doing something against the tumor. It's a bit around the corner, but actually this is progressing quite nicely. So I actually think we really have something there, yeah.
Oh, wow, so it's like a bad B cell subset. Well,
in terms of tumors, yeah, but we are also looking at that in the natural situation, trying to figure out, I mean, they must have a purpose, right? The body doesn't just produce a random B cell subset, which is super specific without it having any use. And it might be something that might, again, help us against autoimmunity. I don't know yet. But it's definitely, for me, it's definitely worth looking deep into that.
Do you get to name your own B cell subset then?
I hope so.
How will you name it?
Well, I didn't really think about it, but I did get to name a mouse strain once, and so I really liked that one. I called them the Humi mice.
Oh,
They were quite cute. Anyway, so we have this B cell subsets, and so it might be the same that is already published as inhibitory B cells, although people don't like that because it's super undefined. They basically just say, B cells, which in this one specific autoimmunity model produce IL-10. So people in the B cell field, like a lot of them don't believe in inhibitory B cells or in regulatory B cells. So I have to see whether I can publish it under either of those names, or whether people will immediately hate it just because I picked a name that is associated with this, in some cases, not great defined cells. Yep. But I actually think those might be similar cells to what we are looking at, but just in this very specific context that they were studied, those studies are not super convincing to people in the B cell field always. But I think if we can properly define them before there is like an autoimmunity, I think this whole story will make way more sense.
Yeah, but with names, there is always this problem in science. It's also happened with macrophages, right? Like M1, M2, then M1-like, M1-2 like, M2-like, at the end. Yeah, that's names in science. It's a bit busy.
And everyone has their own criteria for what that cell is.
Yeah.
Like, I talked to a postdoc the other day, and he was telling me he had a reviewer on his paper telling him that like eosinophils don't exist or something, and I was like, what is even going on? Like, how do you make heads or tails or anything?
It's tricky, and it's really hard to convince people. I mean, there's also now this age-associated B cells that only really came up in meetings and so on a couple of years ago, and which are now a big thing, but again, to very specific models like lupus, for example. So again, yes, you see them coming up with age. Yes, there is an association with very specific autoimmunity or autoimmune diseases, but that also took a while for people to actually be convinced that this exists, and that the fact that that there is just B cells expressing, in this case, for example, TBET and CD11C, means that there is really that this is any meaning, right? And I think we'll face exactly the same thing with ourselves, but as long as the data is convincing, and if some people can reproduce what you're doing, who knows, maybe in 5-10 years this will be one of the completely normal B cell populations that we talk about.
Yeah.
Then let's talk again about this in 5-10 years.
Sure.
Yeah, so it was a very, very, very nice conversation, but we are not finished, because now we finished our teas. So it's time for the last sip question. That is the 3 questions that we ask to our guests always. So they're a bit philosophical, so you can take your time to answer them. And the first one is, if you could go back and speak to yourself on your first day in the lab, what would you say?
Yeah, that's that's not a super straightforward one to me, because actually, although I had all these obstacles, I do believe that I did, from my perspective, things right. I mean, I ended up where I want to be. I'm still doing science. I have my family and everything, so nothing to complain about how my life went, right? So to me, my biggest advice would be, I had a lot of crossroads where I had to choose between going to this lab or that lab, I could have changed career path and go to Boehringer, for example. I had a job offer there and so on. And what I always kept on doing was to just follow my gut feeling about the people that I would work with. So I think it's super hard to judge, especially in the beginning, whether a project is good or bad. So I had several times that people told me this project is on a silver platter, or this will lead to a great publication within a year. And you can't judge whether that's true or not. If you're lucky, you get to see some data. If you're lucky, you understand them enough that you can judge them. But for me, I always looked like, who will I work with? I mean, especially, of course, first the supervisor, that has to work out. You have to be able to communicate with the supervisor. Otherwise, you will have a very hard time. But also just general things. Do I feel welcome in the lab? Do I have the feeling that they do things together? Do I have the feeling that there is this kind of community feeling? Because it's so important that you have this structure which can support you when stuff is not working.
Yep.
It was especially important when I went to Vienna, because there you start from scratch, you don't know anybody. And having a support structure is so essential. And the same here, I just really like the people I'm working with. So you go and you say, hey, I have a super busy experiment today, and then 5 people will offer to help you. And then the other way around, you go and help them, and it's just this communal feeling.
The second question, it will be, what's one mystery, scientific or personal, that you hope to see solved in your life?
Not really a mystery per se, and it's a super egoistic answer. But my child has diabetes type 1 and celiac disease. And the combination is so, let's just call it annoying. I mean, celiac disease is mainly like you want to go somewhere and you want to get food for your child. They go like, what can he eat on the menu? Oh, there is fries, but they might have been just put into wheat because that's where they often are, so that they don't stick together. Or they might have been in the same air fryer, God knows what, like the schnitzel or whatever.
Yeah.
So, this is an annoying disease, but of course it's not immediately life-threatening. The diabetes requires a lot of, getting up in the middle of the night because the sugar is at 300, or because the sugar is down to the lowest possible measurement point, which is 39, which is at one point really getting life-threatening. So you get up and you give the kid sugar in the middle of the night, which always be great. You wake up to drink your apple juice and so on. And just getting a cure for either of the two would be so extremely nice. And of course, especially the diabetes is so common and I think so often overlooked that it has like an incidence rate of 1%. So the chances that your child will have diabetes is 1 in 100, which is a lot.
Yeah.
But I think it's so not present in our society. And there is a lot of things that make it way easier than it was years ago when it comes to sensors and so on. But there is no obvious cure because the cells which produce the insulin are gone. You could put the cells back potentially, but then again, you have the immune system which will target the cells. So you can only put them back if you shut down the immune system, which is of course not a cure. I mean, this is like, it's fighting something bad with something potentially even worse. So for me, solving how to stop the immune system from targeting In this case, they are called beta cells, insulin-producing cells. And at the same time, having a way to safely put the cells in, that would be the one big mystery. I was settled for the celiac disease if I can't get the other one. And diabetes would be my preferred mystery.
Yeah. I mean, I don't think it's selfish. I think there is, as you said, 1% of the population has it. So
Yeah, I think
for sure. And then the last question is, if you could ask the immune system one question and get a clear, honest answer, what would you ask?
I think it goes into exactly the same direction.
Mm-hmm.
Like, why the hell do you think it's a good idea to go against those factors which you need for the body to survive? Why would you target insulin? I mean, of all things, I mean, of course, you ask the question with every single autoimmune disease.
Mm-hmm.
That one is really weird to me. I mean, at least with the wheat, it's the stuff that comes from the outside, which is bound, and the target receptor, which are bound. But this makes more sense to me than to say, hey, I just randomly destroy those specific cell type, which is only in one place in the body, right? It's better. And to say, hey, those ones we don't need, is really like, what are you thinking, immune system?
Yeah. I mean, I think there are two very fair questions, like your answers at the end. So very good. So thank you very, very much for coming, for letting us interview you. It was very, very nice.
Yeah.
Yeah, to me as well. Thank you so much.
So stay curious
Stay critical.
and see you next time for some tea.
Reach us on Instagram at IMmuneFAU or send us questions, ideas, or feedback to immunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunk Studio and Thomas Bauernschmidt.